In the most remote corners of the Siberian taiga, there were cities that did not appear on maps. Scientific centers where Soviet scientists spent decades creating weapons capable of destroying all life on the planet. Biological laboratories whose existence was categorically denied. What was happening behind the barbed wire of these ghost towns? What deadly secrets does the Siberian land still hold? And most importantly, are all these programs really a thing of the past? Today, we will lift the veil on one of the most secret pages of history. If this material has made you think about the hidden pages of history, be sure to subscribe to the channel and like the video. Ahead of us lie new investigations into secrets that the authorities prefer to keep hidden. Now, write your opinion in the comments. Could the technologies from Siberian biolaboratories have fallen into the hands of terrorists after the collapse of the USSR? Share your theories. Truth is born in discussion. In the heart of Siberia, among endless forests and snow-covered plains, lie cities that are not marked on public maps. These are scientific centers surrounded by barbed wire and strict secrecy. These are places where Soviet scientists worked on creating weapons capable of destroying all life on the planet. These are biological laboratories whose existence was denied by the authorities for many years. Today, we will lift the veil of secrecy on one of the Soviet Union's most classified programs. A program to develop biological weapons that encompassed dozens of research centers from the Urals to the Far East. But why did Siberia become the center of this sinister research? The story begins in the mid-20th century, when the world was still reeling from the horrors of World War II. The Soviet leadership understood that future conflicts would require fundamentally new weapons. Not nuclear weapons, whose traces are too obvious, but biological weapons, invisible, silent, but deadly. The choice of Siberia was no accident. The vast distances between settlements provided natural isolation. The harsh climate created additional barriers for the curious. Most importantly, it was possible to conduct tests over large areas without fear of unwanted witnesses. The first secret laboratories appeared as early as the late 1940s. Officially, they were engaged in peaceful research, studying natural infections, developing vaccines, and fighting epidemics. In reality, scientists were working on creating strains of deadly bacteria and viruses capable of overcoming any protective barriers of the human body. What was happening behind the fences of these scientific towns? Documents declassified after the collapse of the USSR reveal only a small part of the truth. Most of the archives remain inaccessible, and many participants in the program took their secrets to the grave. It is known that Soviet biologists studied the military applications of anthrax, plague, smallpox, cholera, and dozens of other deadly diseases. They experimented with genetic modifications in an attempt to create pathogens resistant to antibiotics and vaccines. They worked on aerosol forms of biological agents. Particular attention was paid to the development of so-called ethnic weapons, pathogens capable of affecting people of certain racial groups. This program, codenamed Genome, was one of the most secretive even within the biological program. The scale of the research is staggering. Thousands of scientists, engineers, and technicians participated in the program. Entire scientific cities were built with their own infrastructure: housing, schools, hospitals, and shops. A parallel reality was created, existing according to its own laws. The residents of these towns lived in conditions of triple isolation. The first barrier was the secrecy of the research itself. The second was their geographical remoteness from major centers. The third was a regime of strict secrecy, where even the scientists' families were not allowed to know the nature of their work. But what price did these people pay for participating in the secret program? Many of them suffered from occupational diseases associated with working with dangerous pathogens. Statistics on morbidity and mortality among biological laboratory employees are still classified. Even more tragic was the fate of the test animals. Tens of thousands of monkeys, dogs, rabbits, and mice died during the experiments. At secret testing grounds in the Siberian taiga, biological agents were tested on large animals such as horses, cows, and deer. Today, many of these laboratories have been officially repurposed for peaceful research. But have all military programs really been shut down? An analysis of recent publications by Russian biologists, patent applications, and international contracts suggests that some developments are continuing under the guise of medical research. This story is not just a trip down memory lane. It helps us understand the current realities of biological security and the threats that may arise in the future. The idea of creating biological weapons in the USSR did not come out of nowhere. Back in the 1920s, Soviet military leaders studied the use of chemical weapons in World War I. Realizing the limitations of chemical agents, they began to look for alternatives. Biological weapons seemed like the ideal solution: cheap to produce, invisible when used, and extremely effective. The first official research began in the 1930s as part of the activities of the Kirov Military Medical Academy. A group of scientists led by microbiologist Yakov Rapoport studied the possibilities of military use of pathogenic microorganisms. The work was carried out in strict secrecy under the guise of a program to combat infectious diseases. But what made the Soviet leadership so actively develop this area? The answer lies in intelligence data on similar programs in Nazi Germany and Japan. The infamous Unit 731 of the Japanese Imperial Army was already conducting horrific experiments on humans in Manchuria. The Soviet Union could not afford to fall behind in this deadly race. The Great Patriotic War temporarily halted biological research. All efforts were focused on the creation of traditional weapons. However, in 1944, when victory became apparent, the program resumed with renewed vigor. This time, not only Soviet scientists were involved in the work, but also captured German specialists. Among the trophies brought back from Germany were not only rocket technology and nuclear developments, but also documents on the Third Reich's biological programs. German scientists had been working on the creation of bacteriological bombs, aerosol methods of spraying pathogens, and genetic experiments. These materials became the basis for Soviet research. The program was formally established in 1946 with the creation of the 15th Main Directorate under the USSR Ministry of Defense. Officially, it was responsible for chemical and biological defense. In reality, this agency coordinated all work on the creation of biological weapons. Why was Siberia chosen? In addition to its geographical advantages, there were other factors. The region already had major scientific centers: Novosibirsk Akademgorodok, Tomsk Polytechnic Institute, and Irkutsk University. The best minds of Soviet science were concentrated here. Another important factor was the proximity to natural sources of particularly dangerous infections. Siberia and the Far East are the natural habitats of the pathogens that cause plague, tularemia, anthrax, and tick-borne encephalitis. Studying these pathogens in their natural environment provided unique opportunities for the creation of biological weapons. The first major scientific center was established in Koltsovo, near Novosibirsk, in 1951. Officially, it was called the Institute of Molecular Biology, and later the State Scientific Center for Virology and Biotechnology, Vector. But what kind of research was actually conducted there? Documents testify to work with smallpox, Marburg, Ebola, and Lassa viruses. Soviet virologists attempted to create strains resistant to existing vaccines. They studied the possibilities of aerosol infection and developed methods for the mass production of viral agents. At the same time, a center for the study of bacterial pathogens was being created in Omsk. Here, they worked with the pathogens that cause plague, anthrax, tularemia, and cholera. Particular attention was paid to creating strains with multiple drug resistance. The program developed in several directions simultaneously. The first was the creation of new pathogenic strains with improved combat characteristics. The second was the development of means of delivering biological agents to their targets. The third was the creation of systems for the mass production of biological weapons. The fourth was ensuring the protection of its own troops from enemy biological attacks. By the end of the 1950s, dozens of scientific institutes, design bureaus, and manufacturing enterprises were participating in the program. The total number of personnel involved reached several tens of thousands. Funding was provided from closed items in the defense budget and amounted to billions of rubles per year. What were the goals set for the creators of biological weapons? Analysis of declassified documents shows that the plan was to create an arsenal for large-scale biological warfare. The idea was to destroy enemy cities, industrial centers, and military facilities using aerosol forms of pathogens. Plans for the diversionary use of biological weapons occupied a special place. Means of covertly infecting water supply systems, food warehouses, and livestock farms were developed. Portable devices were created to infect enclosed spaces such as subways, office buildings, and airplanes. The international situation contributed to the development of the program. The Cold War required the creation of fundamentally new types of weapons. Biological weapons seemed to be the answer to the nuclear superiority of the United States. But did the creators understand the danger they posed to all of humanity? In the 1960s, Novosibirsk's Akademgorodok was a showcase for Soviet science. Created as a center for advanced research, it attracted the country's best scientists. Among the pine forests beyond the Ob Sea, a unique scientific city grew up, where fundamental science coexisted with secret military developments. But did the residents of Akademgorodok know that one of the most dangerous scientific centers on the planet was being created 20 kilometers away from them? The village of Koltsovo appeared on maps in 1952 as Post Office Box number 230. The official goal was to create an institute for the study of natural focal infections in Siberia. In reality, work began here on the creation of a new generation of viral biological weapons. The choice of location was not accidental. Koltsovo was close enough to Novosibirsk to use the scientific base of Akademgorodok, but far enough away to ensure secrecy. Tolmachevo Airport allowed for the rapid delivery of samples and specialists. The proximity to the railway facilitated the transportation of equipment. The first director of the institute was Lev Sandachev, an outstanding Soviet virologist and student of the famous Vitaly Zhdanov. Under his leadership, the small laboratory became the world's largest center for the study of particularly dangerous viruses. What made this talented scientist devote his life to the creation of biological weapons? Sandichev's official biography is full of gaps. It is known that he participated in the smallpox eradication program, worked with exotic African viruses, and had close ties to military circles. But his real role in the creation of the Soviet biological arsenal remains a mystery to this day. In the 1960s, construction began on a modern scientific complex in Koltsovo. Outwardly, it was no different from ordinary academic institutions. Several glass and concrete buildings surrounded by a park. But underground were laboratories with the highest level of biological protection, capable of working with the most dangerous pathogens. The security system was impressive in its complexity. Multiple decontamination barriers, autonomous life support systems, special pressure suits. Even the air leaving the laboratories underwent several stages of purification and sterilization. But could these measures completely eliminate the risk of infection for the staff? Accident statistics in Koltsovo are still classified. However, there are several known incidents in which employees became infected as a result of safety protocol violations. In 1977, researcher Nikolai Ustinov died of Marburg fever. His death was the first officially recognized case of occupational infection with a particularly dangerous infection in the USSR. The main research in Koltsovo focused on viruses that cause hemorrhagic fevers, smallpox, and encephalitis. Soviet virologists studied the possibility of creating recombinant strains that combine the properties of various pathogens. Methods were developed to increase the virulence and contagiousness of natural strains. A special focus was placed on working with the natural smallpox virus. Officially, this virus was eradicated worldwide as a result of an international smallpox eradication program. There were only 2 places left on the planet where samples of the virus were stored: the Centers for Disease Control in Atlanta and the Ivanovsky Institute of Virology in Moscow. But did Koltsovo really not work with the smallpox virus? Declassified documents show that Soviet virologists created genetically modified variants of the smallpox virus that were resistant to existing vaccines. This work was carried out as part of the Great Fire Project, one of the most secretive biological weapons programs. In parallel with viral research, work was underway in Koltsovo to develop means of delivering biological agents. Aerosol generators capable of spraying viruses over large areas were designed. The possibility of using missile warheads to deliver biological charges was studied. By the 1980s, the Vector State Research Center for Virology and Biotechnology had become one of the largest virology centers in the world. It employed more than 3,000 people, including leading specialists in molecular biology, genetics, and biotechnology. The center's budget amounted to hundreds of millions of rubles per year. But the true scale of Vector's activities only became known after the collapse of the USSR. It turned out that Soviet virologists not only created biological weapons, but also developed plans for their mass use. What targets were planned to be hit with the viral weapons created in the Siberian taiga? An analysis of captured documents showed that in the event of war, Vector was supposed to ensure the defeat of the enemy's largest cities. Attacks were planned on New York, London, Tokyo, and other megacities. The use of aerosol forms of viruses made it possible to infect millions of people almost simultaneously. The current status of the Vector Center raises many questions. Officially, it has been repurposed for peaceful research, the development of vaccines, diagnostic systems, and medicines. But a regime of heightened secrecy remains in place. with limited access for international inspectors and the classification of many areas of work. Can we be sure that military programs in Koltsovo have been completely shut down? An analysis of patent applications, scientific publications, and international contracts of the center suggests that dual-use research is continuing. Under the codename Factor, Soviet documents concealed one of the most ambitious projects to create biological weapons. This facility, located in the closed scientific center Vector in Koltsovo, was intended for the development of fundamentally new types of viral weapons. What made Factor so special among other biological programs of the USSR? The project began in 1971, after the signing of the Convention on the Prohibition of the Development, Production and Stockpiling of Bacteriological, Biological, and Toxin Weapons. Paradoxically, it was international restrictions that stimulated the development of the Soviet biological program. The country's leadership understood that if biological weapons were banned, it meant that they were truly effective. Factor was an attempt to circumvent international agreements with the help of new technologies. Instead of traditional pathogens, the plan was to create artificial biological agents that were not formally covered by the convention. Genetic engineering opened up unlimited possibilities for the creation of a new generation of weapons of mass destruction. Sergei Netesov, a brilliant molecular biologist and student of the founders of Soviet virology, became the scientific director of the project. Under his leadership, a unique laboratory equipped with the most modern equipment was created. It was here that the first DNA sequencing machines, gene synthesizers, and cell culture systems appeared in the USSR. What specific viruses were studied as part of the Factor project? Declassified documents mention work with the Marburg virus, Lassa fever, hantaviruses, and arenaviruses. Particular attention was paid to African hemorrhagic fevers, diseases with a mortality rate of up to 90%. Soviet virologists obtained samples of exotic pathogens through a network of friendly countries in Africa and Asia. Officially, these were scientific exchanges within the framework of technical assistance programs for developing countries. In reality, a collection of the most dangerous viruses on the planet was systematically assembled. One area of work was the creation of chimeric viruses, artificial pathogens combining the properties of various natural strains. In theory, this approach made it possible to create viruses with specific characteristics: high contagiousness, long incubation period, resistance to treatment. In the mid-1970s, Vector began work on the Hunter project. The goal was to create a virus capable of selectively infecting people of certain ethnic groups. The basis for this was the differences in the structure of receptor proteins in representatives of different races. How realistic were the plans to create an ethnic biological weapon? Modern genetic research shows that the differences between races are too insignificant to create a selective pathogen. However, in the 1970s, knowledge of the human genome was insufficient to understand these limitations. At the same time, work was underway to create binary biological weapons. The idea was to use two relatively safe components that would only become lethal when combined in the victim's body. This approach made it possible to circumvent biological defense systems and international controls. Research into the creation of viruses resistant to existing vaccines and drugs occupied a special place in the Factor project. To this end, methods of directed evolution were used, the artificial selection of strains with the desired properties. The process was accelerated by mutagenic effects and genetic manipulation. By the early 1980s, Vector had created a unique collection of dangerous viruses. In addition to natural strains, dozens of artificially modified variants were stored there. What security measures were used to store this deadly arsenal? Virus cultures were kept in special cryogenic storage facilities at a temperature of -70 degrees Celsius. Only employees with the highest level of clearance had access to them. Each sample was registered in a multi-level accounting and control system. Periodic inventories of the collection were conducted. Despite all the precautions, the security system had vulnerabilities. In 1979, there was a leak of a hemorrhagic fever strain, which led to the infection of laboratory animals in a neighboring building. The incident was successfully concealed, but it revealed the imperfection of the existing protocols. The international situation in the 1980s contributed to the further development of Project Factor. The deterioration of relations between the USSR and the US, the launch of Reagan's Star Wars program, and the war in Afghanistan all required the creation of new types of weapons. Was the practical application of viruses created as part of Project Factor planned? Analysis of military documents shows that scenarios for the use of biological weapons in various conflicts were developed. Options for sabotage operations, striking enemy rear facilities, and creating epidemics in cities were considered. The collapse of the Soviet Union raised the question of the fate of the pathogens accumulated at Vector. Part of the collection was destroyed under international supervision. But can we be sure that all dangerous strains have really been eliminated? The current status of the Factor project remains a mystery. Officially, all military research at Vector has been discontinued. The center is engaged in the development of vaccines, diagnostic systems, and fundamental research in the field of virology. But the regime of secrecy remains, and access for international inspectors is limited. During the Soviet era, Omsk was not only a major industrial center in Siberia, but also one of the key hubs of the biological weapons program. Here, in the secret laboratories of military institutes, bacterial agents of mass destruction were developed. What was hidden behind the facade of peaceful scientific institutions in this Siberian city? The history of military biological research in Omsk began during the Great Patriotic War. In 1942, some of the scientific institutes from the European part of the USSR were evacuated here. Among them were laboratories engaged in the study of particularly dangerous infections. The war required means of protection against the possible use of biological weapons by the enemy. After the war, these temporary laboratories became permanent research centers. In 1947, the Omsk Research Institute for Plague Control was established. Officially, it was engaged in the study of natural foci of plague in western Siberia and the development of preventive measures. In reality, the institute became one of the centers of the Soviet biological weapons program. The first director of the institute was Mikhail Timofeyev, an outstanding epidemiologist who had participated in the fight against epidemics in Central Asia. Under his leadership, the modest laboratory was transformed into a large scientific complex. But what kind of research was being conducted behind the closed doors of the Omsk laboratories? The main focus was on studying the causative agent of the plague, Yersinia pestis. This bacterium, which had caused devastating pandemics in the past, was considered an ideal biological weapon. Its high lethality, ability to spread through aerosols, and ability to survive in the environment made the plague attractive for military use. Soviet microbiologists studied various strains of the plague bacillus isolated from natural foci in Siberia and Central Asia. Of particular interest were strains with increased virulence, capable of overcoming natural human immunity. Experiments were conducted to artificially increase the pathogenicity of the bacteria. At the same time, work was carried out on the causative agent of anthrax, Bacillus anthracis. This bacterium forms spores that can survive in the environment for decades. The aerosol form of anthrax was considered one of the most effective types of biological weapons. What methods were used to create combat strains of bacteria? In the 1950s, the main method was the selective cultivation of the most aggressive variants. The bacteria were exposed to various mutagenic factors, radiation, chemicals, extreme temperatures. The surviving strains often acquired new dangerous properties. With the development of genetics in the 1960s, more sophisticated methods of modifying bacteria appeared. Soviet microbiologists learned to embed additional virulence genes borrowed from other microorganisms into the genome of pathogens. Chimeric strains with combined properties were created. A special focus was placed on developing bacteria resistant to antibiotics. Already in the 1950s, it was clear that the enemy would use antibacterial drugs to treat the infected. The creation of resistant strains made it possible to overcome this defense. Tests of aerosol forms of bacterial agents were conducted in underground laboratories in Omsk. The survival of microorganisms when sprayed and their ability to overcome the protective barriers of the human respiratory system were studied. Optimal particle sizes were developed for maximum penetration into the lungs. How dangerous were these experiments for the researchers themselves? Despite precautions, cases of occupational infection occurred regularly in the Omsk laboratories. The statistics are classified, but several fatalities among the institute's employees are known. In 1964, a serious incident occurred that nearly led to a plague epidemic in Omsk. Due to a violation of safety protocols, laboratory animals infected with an experimental strain came into contact with urban rodents. Only emergency pest control measures prevented the spread of the infection. By the 1970s, the Omsk Anti-Plague Institute had become one of the leading centers for the study of biological weapons in the USSR. Hundreds of specialists work there, including microbiologists, epidemiologists, and biotechnology engineers. Production facilities were created for the industrial-scale production of combat strains. The institute worked closely with military design bureaus involved in the creation of biological weapon delivery systems, special ammunition for missiles and artillery, aerosol generators for aircraft, and portable devices for sabotage were developed. What were the plans for the combat use of Omsk's developments? Analysis of military documents shows that bacterial weapons were considered a strategic tool. Massive strikes on the enemy's largest cities were planned with the aim of creating uncontrollable epidemics. Particular attention was paid to the destruction of military facilities, bases, warehouses, ports, and airfields. Bacterial agents made it possible to disable enemy personnel without destroying material assets. Contaminated areas became unusable for a long time. The 1972 International Convention on the Prohibition of Biological Weapons formally halted military research in Omsk. But were all programs really shut down? The Institute continued its work under the guise of studying natural focal infections and developing means of protection against bioterrorism. The current state of the Omsk Institute raises questions. A regime of heightened secrecy remains in place, and access for international observers is restricted. Research with particularly dangerous pathogens continues under the guise of biological safety programs. Could the developments of Omsk scientists have fallen into the hands of terrorists or other states? In the 1990s, amid the collapse of science funding, many specialists left the institute. Some of them emigrated to other countries, taking with them unique knowledge and technologies. Krasnoyarsk 26 did not exist on the official maps of the Soviet Union. This closed city, built deep in the Siberian taiga, was one of the most secret facilities in the country. Officially, plutonium for nuclear weapons was produced here. But what other deadly secrets were hidden behind the barbed wire of this ghost town? The history of Krasnoyarsk 26, later renamed Zheleznogorsk, began in 1948 with the creation of the first industrial reactor in the USSR for the production of plutonium. The city was built by Gulag prisoners under conditions of strict secrecy. Even the builders did not know the true purpose of the facility. The location was chosen for a reason. Its remoteness from major centers ensured secrecy. Its proximity to the Yenisei River provided an unlimited source of water for cooling the reactors. The railway allowed materials to be delivered and finished products to be transported out. Most importantly, it was possible to conduct any experiments here without fear of prying eyes. In the 1950s, along with the nuclear program, the development of biological weapons began in Krasnoyarsk 26. The combination of radiation and biological research seemed logical. Both required maximum secrecy and special security measures. Why was the nuclear center chosen for biological research? The answer lies in the unique opportunities offered by the presence of research reactors. Radiation exposure made it possible to create mutant strains of microorganisms with specific properties. The process of mutagenesis was significantly accelerated compared to natural conditions. The biological department of Krasnoyarsk 26 was created as part of a program to develop radiological weapons, a combination of radioactive isotopes and pathogenic microorganisms. Such weapons combined immediate radiation damage with delayed biological effects. The main research focused on studying the effects of radiation on pathogenic bacteria and viruses. Scientists discovered that some microorganisms not only survive high doses of radiation, but also acquire new dangerous properties. Their resistance to antibiotics increases, their virulence changes, and new ways of transmitting infection appear. Particular attention was paid to the creation of radiation-resistant strains of pathogens that cause particularly dangerous infections. Such microorganisms could survive in areas contaminated by radiation, posing an additional threat to enemy troops. What experiments were conducted in the underground laboratories of Krasnoyarsk 26? Declassified documents describe work with irradiated strains of anthrax, plague, and tularemia. The possibility of creating binary weapons, a combination of radioactive materials and biological agents, was studied. In the 1960s, the development of so-called dirty biological bombs began. The idea was to spray radioactive dust contaminated with pathogenic microorganisms. Such weapons created areas of long-term contamination, unsuitable for life and economic activity. At the same time, the possibility of using radioactive isotopes to sterilize biological agents was being studied. This made it possible to create vaccines from pathogens that had been killed by radiation but retained their antigenic properties. This approach accelerated the development of defenses against biological weapons. Experiments with transuranic elements, plutonium, americium, and curium, posed a particular danger. These substances are not only highly radioactive, but also chemically toxic. Their combination with pathogenic microorganisms created a triple-action weapon: radiological, chemical, and biological. What safety measures were used when working with radioactive pathogens? The laboratories were located in underground bunkers with a multi-level protection system. In addition to biological isolation, protection from radiation was required. The staff worked in special suits equipped with autonomous life support systems. Despite the precautions, serious accidents occurred at Krasnoyarsk 26. In 1962, there was a leak of a radioactive strain of anthrax, which led to the contamination of a large area of taiga. The exclusion zone still exists today. By the 1970s, the radiobiological weapons program had reached its peak. An experimental reactor was built in Krasnoyarsk 26 specifically for irradiating biological cultures. Its capacity allowed it to process tons of pathogenic material per year. The International Convention on the Prohibition of Biological Weapons formally halted this research. But were all programs really shut down? An analysis of the current state of Zheleznogorsk shows that dual-use work is continuing. Today, the Federal Nuclear Center, which deals with the disposal of nuclear materials, operates on the territory of the former Krasnoyarsk 26. But the closed city regime remains in place, and access for international inspectors is restricted. Officially, all biological research has been discontinued. But can this be believed? Current satellite images show the construction of new facilities on the center's territory. Analysis of open sources reveals the continuation of research in the field of radiobiology. Works on the effects of radiation on microorganisms are being published, and new laboratories with enhanced biological protection are being created. What threat do the radioactive materials stored in Zheleznogorsk pose? In addition to plutonium, tons of other transuranic elements are stored here. If these materials fall into the hands of terrorists, they could be used to create a new generation of radiological weapons. The fate of the biological collections accumulated over decades of research is particularly alarming. Officially, all dangerous strains have been destroyed or transferred to other research centers. But given the scale of previous programs, can we be sure that this legacy of the Cold War has been completely eliminated? In the most remote corners of the Siberian taiga, where maps show only white spots, there were secret testing grounds for biological weapons. Here, across thousands of square kilometers of wilderness, the Soviet military tested the lethal effectiveness of pathogens created in laboratories. What happened in these death zones, hidden from the eyes of the world? The first large biological testing ground was created in 1952 on Vozrojdeniya Island in the Aral Sea. But the harsh conditions of Siberia offered unique opportunities for larger-scale testing. Low temperatures allowed researchers to study the survivability of pathogens in extreme conditions. Vast uninhabited territories ensured complete isolation for the experiments. The main Siberian testing ground was located in the Evenk Autonomous Okrug, near the village of Baykit. Officially, this territory was considered a hunting reserve. In reality, it was the site of the most dangerous experiments of the Soviet biological program. The testing ground was given the code name Alabaster. What kind of tests were conducted in the Siberian wilderness? Declassified documents describe experiments involving the spraying of aerosols containing anthrax, plague, and tularemia over areas of taiga covering several thousand hectares. The range of the infectious clouds, their resistance to weather conditions, and their effectiveness in hitting living targets were studied. The main test subjects were wild and domestic animals. Thousands of cattle, horses, pigs, and sheep were brought to the test site. Simulations of livestock farms and military camps were created. After the biological agents were sprayed, the development of the epidemic process was monitored. Particular attention was paid to testing means of delivering biological weapons. Special rockets, artillery shells, and aircraft bombs were developed for spraying pathogens. Aerosol generators of various capacities were created, ranging from portable devices to aircraft systems for mass use. How was the secrecy of the tests ensured in open terrain? The test site was guarded by several rings of military posts. The outer perimeter was controlled by border guards, and the inner perimeter by special units of the GRU. Any economic use of the territory within a radius of 100 kilometers was prohibited. The local population, the Evenks and Yakuts, were forcibly resettled to other areas under the pretext of creating a nature reserve. Reindeer pastures were declared infected with an unknown livestock disease. Hunting and fishing in the region were completely banned. To simulate real combat conditions, models of city blocks, military bases, and industrial facilities were created at the test site. Real buildings were used, transported from areas flooded by Siberian hydroelectric power plants. Test animals were placed in these settings to study the penetration of biological agents into shelters. Winter tests were of particular importance. At temperatures of -30 degrees, most pathogens die quickly. But Soviet scientists developed special cryoprotectants, substances that protect bacteria and viruses from freezing. Strains adapted to Arctic conditions were created. How effective were the tested samples of biological weapons? According to declassified reports, the aerosol form of anthrax affected 95% of the test animals when sprayed from an airplane at an altitude of 500 meters. The mortality rate reached 80% within a week after infection. Experiments with primates were particularly secret. Hundreds of monkeys were brought to the test site from nurseries in Sukhumi and Adler. These animals, genetically close to humans, made it possible to model the effects of biological weapons on humans without direct testing on humans. The results of the experiments with primates showed the monstrous effectiveness of the biological agents created. Modified strains of anthrax killed monkeys within 2 to 3 days. The symptoms resembled lightning pneumonia with massive internal bleeding. By the 1970s, more than 100 different strains of pathogenic microorganisms had been tested at the Alabaster test site. Detailed tables were created showing the combat effectiveness of each agent in different climatic conditions and methods of application. What was the environmental cost of these tests? Tens of thousands of square kilometers of Siberian taiga were contaminated with anthrax spores and other pathogens. Many areas remain dangerous to this day. Anthrax spores remain viable in the soil for decades. The mass death of wild animals in the test site area was attributed to natural epidemics and harsh winters. But analysis of archival documents shows abnormally high mortality rates among deer, bears, and fur-bearing animals within a 200-kilometer radius of the test center. Local residents preserved oral traditions about the years of plague when entire animal populations died out. Evenki elders told of dead lakes where fish no longer lived, and cursed glades where grass no longer grew. Could they have known the true cause of these disasters? The closure of the test site began in 1975, after the signing of international agreements to limit biological weapons testing. But the process dragged on for decades. The last experiments were conducted in the 1980s under the guise of biological defense research. The current state of the former Alabaster test site remains a mystery. The area is still closed to visitors. Satellite images show destroyed buildings and overgrown test sites. But have the consequences of decades of deadly experiments been completely eliminated? Environmentalists have detected abnormal concentrations of anthrax spores in the region's soil. Cases of animal diseases of unknown etiology are periodically recorded. The veterinary service attributes them to natural infections, but the symptoms resemble those caused by experimental strains. What threat do the contaminated areas pose today? Global warming is causing the permafrost to melt, where preserved pathogens may be stored. Climate change could lead to the release of the deadly legacy of the Soviet biological program. The question of responsibility for environmental damage remains open. Russian authorities deny the existence of biological testing grounds in Siberia. The international community does not have access to contaminated areas for an independent assessment of the scale of the problem. In the depths of Siberian laboratories, one of the most sinister projects in human history was born: the creation of genetic weapons capable of destroying people on the basis of race. The Genome Program, launched in the 1970s, was supposed to be the crowning achievement of Soviet biological science. But how real was the threat of creating weapons targeted at specific ethnic groups? The idea of ethnic biological weapons arose after the discovery of genetic differences between races. In the 1960s, scientists discovered that people from different ethnic groups react differently to the same drugs. Some drugs are effective for Caucasians but toxic to Mongoloids. These differences led military biologists to consider the possibility of creating selective biological weapons. The scientific center of the Genome Program was a secret department of the Institute of Cytology and Genetics of the Siberian branch of the Russian Academy of Sciences in Novosibirsk. Officially, they studied the genetics of the populations of the peoples of the USSR. In reality, a group of scientists led by academician Dmitry Belyaev worked on deciphering the genetic codes of various ethnic groups. Which peoples were the subjects of this research? The priority targets were the Chinese, Japanese, Arabs, Jews, and African Americans. Detailed genetic maps were compiled for each group, and unique markers characteristic only of representatives of a particular race were identified. Genetic material was collected under the guise of medical and anthropological research. Soviet expeditions to friendly countries in Asia and Africa systematically collected blood and tissue samples from the local population. Genetic databases of various ethnic groups around the world were created. At the same time, the mechanisms of hereditary predisposition to various diseases were studied. It turned out that some peoples have genetic resistance to certain infections, but increased susceptibility to others. This knowledge was to become the basis for the creation of race-specific pathogens. How successful were the attempts to create ethnic weapons? Documents show that by the early 1980s, Soviet geneticists had identified several potential targets for selective biological weapons. Particular attention was paid to differences in the structure of receptor proteins and enzyme systems. One area of focus was the development of toxins that selectively target people with certain genetic polymorphisms. Plant and animal toxins were studied, the effectiveness of which depended on the genetic characteristics of the victim. A more ambitious task was the creation of viruses capable of selectively attacking cells with a specific genetic profile. To this end, genetic engineering methods that had only appeared in the 1970s were used. Viruses were modified so that they could only reproduce in cells of a specific genetic type. What ethical problems did the participants in the program face? Many scientists understood the monstrosity of their work. The creation of racial weapons contradicted the basic principles of humanism and scientific ethics. Some researchers sabotaged the work by providing deliberately false results. The issue of testing the agents being created was particularly painful. Animal experiments could not provide complete information about the effectiveness of ethnic weapons. Tests on people of different racial types were required. Were such experiments carried out in Soviet laboratories? No direct evidence of human testing has been found. However, an analysis of medical statistics from the 1970s and 1980s reveals abnormal outbreaks of rare diseases among certain ethnic groups in the USSR. The incidence was particularly high among the peoples of Central Asia and the Caucasus. The international situation contributed to the development of the Genome Program. The deterioration of relations with China, the conflict on Damansky Island, and the threat of nuclear war all required the creation of fundamentally new types of weapons. Ethnic biological weapons seem to be the ideal means of defeating the Chinese population of many millions. Scenarios for the use of genetic weapons in various military conflicts were developed. Sabotage in large enemy cities, contamination of water supply systems, and spraying from aircraft over enemy territory were planned. But did military strategists understand the long-term consequences of using such weapons? Modern genetics shows the utopian nature of the idea of creating race-specific biological weapons. Genetic differences between races are significantly smaller than differences within racial groups. Any pathogen targeting one ethnic group will inevitably affect representatives of other races. But in the 1970s, knowledge of the human genome was insufficient to understand these limitations. Soviet scientists sincerely believed in the possibility of creating selective biological weapons. Millions of rubles were spent on research that was obviously futile. What happened to the participants of the Genome Program? After the collapse of the USSR, many of them emigrated to the US, Israel, and other countries. Some continued to work in the field of medical genetics. Some specialists retrained in other fields of science, trying to forget their involvement in the creation of racial weapons. The documents of the Genome Program are still classified. The Russian authorities deny the existence of work on the creation of ethnic biological weapons in the USSR, but declassified materials from other countries confirm the reality of this research. Do modern advances in genetics pose a new threat of creating racial weapons? The Human Genome Project, completed in the 2000s, provided a detailed map of human genes. Gene editing technologies open up fundamentally new possibilities for the creation of biological weapons. Today, it is theoretically possible to create pathogens that target people with specific genetic markers. But such weapons could be directed not against races, but against families, clans, or even individuals. Personalized biological weapons could become a reality in the coming decades. During the Soviet era, Irkutsk was not only the largest city in eastern Siberia, but also one of the key centers for the study of particularly dangerous infections. Here, within the walls of the Anti-Plague Institute, Soviet scientists worked to transform humanity's oldest enemy into a modern weapon of mass destruction. What secrets were hidden behind the facade of a peaceful medical institution on the banks of the Angara River? The Irkutsk Research Anti-Plague Institute of Siberia and the Far East was established in 1934 to study natural foci of plague in the Asian part of the USSR. The region was endemic for plague, with regular outbreaks of the disease among rodents and the population. Initially, the institute was indeed engaged in peaceful research. Scientists studied the ecology of the plague microbe and developed methods for diagnosing and preventing the disease. Anti-plague vaccines and serums were created. But by the end of the 1930s, the nature of the work began to change. What turned a peaceful scientific institution into a center for the development of biological weapons? The answer lies in the characteristics of the plague pathogen. Yersinia pestis has a unique combination of properties that make it an ideal agent for biological warfare. It is highly contagious, capable of aerosol transmission, has a rapid course, and is highly lethal if left untreated. In the 1940s, in parallel with anti-plague activities, the development of combat strains of the plague microbe began in Irkutsk. A special laboratory with enhanced biological protection was created, where experiments with pathogenic cultures were carried out. Particular attention was paid to the study of the pulmonary form of plague, the most dangerous type of the disease. Pulmonary plague is rare in natural infections. However, it poses the greatest threat as a biological weapon due to its ability to be transmitted by airborne droplets. What methods were used to create combat strains of plague? Soviet microbiologists studied various natural variants of the plague microbe isolated in Siberia, Central Asia, and Mongolia. The most virulent strains were selected for further modification. Selective selection and artificial mutagenesis methods were used. The bacteria were exposed to radiation, chemical mutagens, and extreme temperatures. The surviving cultures often acquired new dangerous properties, increased resistance to antibiotics, altered antigenic structure, and enhanced virulence. In the 1950s, a unique bank of plague microbe strains was created in Irkutsk. The collection included samples from all known natural foci on the planet. Of particular value were historical strains preserved from the great pandemics of the past. Among them were cultures believed to originate from the pathogens of the Black Death of the 14th century and the Third Plague Pandemic of the 19th and 20th centuries. Could these ancient strains possess special properties lost by modern variants of the pathogen? In parallel with the creation of combat strains, methods for their mass production were developed. Fermentation facilities for growing plague cultures on an industrial scale were set up in the basement of the institute. The productivity allowed hundreds of kilograms of biomass to be obtained per week. A special area of focus was the development of aerosol forms of plague weapons. Special preparations were created to preserve the viability of bacteria when sprayed into the atmosphere. The optimal particle sizes for maximum penetration into the human lungs were studied. Were plague weapons tested on living subjects? No documentary evidence of experiments on humans has been found. However, tests on laboratory animals, mice, guinea pigs, rabbits, and monkeys are known to have been conducted. The results showed the extreme effectiveness of the preparations created. In the 1960s, microbiologists in Irkutsk began working on the creation of plague strains resistant to streptomycin, the main antibiotic used to treat plague. Such variants of the pathogen rendered existing methods of treatment useless, turning plague into a completely fatal disease. At the same time, they studied the possibility of creating vaccine-resistant strains. Existing anti-plague vaccines only provided protection against natural variants of the pathogen. Artificially modified strains could overcome vaccine immunity. By the 1970s, the Irkutsk Anti-Plague Institute had become one of the world's leading centers for the development of plague biological weapons. Hundreds of specialists worked there: microbiologists, epidemiologists, and biotechnologists. Equipment for the production and use of biological agents was created. What were the plans for the combat use of plague weapons? An analysis of military documents shows that plague was considered a strategic weapon for destroying large enemy cities. Aerosol attacks on megacities with populations of millions were planned. The plague was particularly attractive because of its ability to spread epidemically. One successful attack could lead to an uncontrollable pandemic covering entire continents. The economic and psychological damage from such a strike would be enormous. The International Convention banning biological weapons formally halted military research in Irkutsk. The institute returned to studying natural sources of plague and developing preventive measures. But can we be sure that military programs have been completely discontinued? The current state of the Irkutsk Institute raises questions. A regime of heightened secrecy remains in place, and access for international inspectors is restricted. Research on virulent strains of plague continues under the pretext of developing means of protection against bioterrorism. What threat does the collection of plague strains accumulated in Irkutsk pose? Decades of research have created a unique arsenal of pathogenic cultures. If these materials fall into the hands of terrorists or hostile states, they could become the basis for the creation of a new generation of biological weapons. Sverdlovsk, 1979. The city was suddenly hit by a wave of deadly diseases. People were dying from a mysterious fever with symptoms of severe pneumonia. The official version spoke of an outbreak of intestinal anthrax sepsis caused by the consumption of contaminated meat. But what really happened in the Ural city in the spring of 1979? Sverdlovsk 19, later renamed Yekaterinburg, was one of the largest centers of the Soviet defense industry. Rockets, tanks, and chemical weapons were manufactured here. But few people knew that a secret facility for the production of biological weapons was located in the industrial area of the city. Military Town Number 19 did not officially exist on maps. This classified complex was created in the 1950s to produce combat strains of anthrax. The plant was given the code name Compound 19 and was part of the 15th Main Directorate of the USSR Ministry of Defense. Why was anthrax chosen as the main biological agent in the Soviet arsenal? The pathogen that causes this disease, Bacillus anthracis, has unique properties. It forms spores that can survive in the environment for decades. The aerosol form of anthrax causes a rapid lung infection with a mortality rate close to 100%. Production began in 1952 with the creation of an experimental facility capable of producing several kilograms of spores per month. By the 1970s, the plant had become the world's largest biological weapons production facility. Its production capacity allowed it to produce tons of dry anthrax spores annually. The production technology was classified at the highest level. The bacteria were grown in huge fermenters and then dried using a special method to obtain a fine powder. The finished product was packed in sealed containers and sent to warehouses for long-term storage. What security measures were in place at the biological weapons factory? The facility was surrounded by several security rings. Staff underwent thorough screening and were regularly vaccinated against anthrax. The production facilities were equipped with biological protection and emergency decontamination systems. Despite all the precautions, working with pathogenic microorganisms on an industrial scale was extremely dangerous. Minor accidents occurred periodically: pipeline ruptures, filter system failures, equipment leaks. Usually, They were eliminated without serious consequences. But on April 2, 1979, a disaster occurred that could not be concealed. As a result of a technical malfunction, aerosols containing anthrax spores escaped from the plant's ventilation pipe into the atmosphere. The wind carried the deadly cloud toward residential areas of the city. How many people were affected by this man-made disaster? Official USSR data reported 64 deaths, but an analysis of medical records conducted after the country's collapse showed a significantly higher number of victims. The actual number of deaths could have reached 100 to 150 people. The first cases of the disease were recorded on April 4th. People were admitted to hospitals with symptoms of severe pneumonia, high fever, difficulty breathing, and coughing up bloody phlegm. Doctors were unable to make an accurate diagnosis. The clinical picture did not correspond to any known diseases. The Soviet authorities immediately classified information about the outbreak. The patients were isolated in a special infectious diseases ward. Medical staff were given strict instructions not to disclose any details of what was happening. Relatives of the deceased were given death certificates stating that the cause of death was ordinary pneumonia. What measures were taken to eliminate the consequences of the accident? A special commission headed by the Deputy Minister of Health of the USSR arrived in Sverdlovsk. A headquarters was set up to combat the epidemic, and all the city's medical forces were mobilized. Mass preventive vaccination of the population against anthrax was carried out under the guise of flu vaccinations. Potentially contaminated areas were disinfected. Stray animals that could have been carriers of the infection were destroyed. The biological weapons plant was temporarily shut down to investigate the causes of the accident and modernize safety systems. Some of the staff were transferred to other enterprises. The plant's management was subjected to repression. The international community was unaware of the Sverdlovsk disaster for a long time. The USSR categorically denied the production of biological weapons. The official version of food poisoning from contaminated meat was maintained for decades. The truth only came to light after the collapse of the Soviet Union. In 1992, the Russian authorities officially acknowledged the accident at the biological weapons factory. President Yeltsin personally apologized to the families of the victims. What lessons can be learned from the Sverdlovsk tragedy? The accident demonstrated the extreme danger of biological weapons production. Any technical malfunction can have catastrophic consequences for the civilian population. The event also demonstrated the Soviet leadership's willingness to conceal the truth about man-made disasters. The secrecy surrounding the information prevented an adequate medical response and international cooperation in dealing with the consequences. Have the consequences of the Sverdlovsk accident been completely eliminated? Anthrax spores are extremely resistant to external influences. They can remain in the soil for decades. Areas that were contaminated in 1979 may still pose a danger. Recent studies show elevated levels of anthrax spores in the soil of some areas of Yekaterinburg. Cases of disease in animals that have come into contact with contaminated soil are periodically reported. Could there be a renewed outbreak of infection decades after the accident? The Sverdlovsk tragedy has become a symbol of the danger of biological weapons. It showed that such weapons threaten not only the enemy, but also the population of the country that possesses them. The memory of those who died reminds humanity of the need for a complete ban on the development and production of biological weapons of mass destruction. After the signing of the Biological Weapons Convention in 1972, the world breathed a sigh of relief. It seemed that humanity had forever renounced the creation of biological weapons of mass destruction. But did Soviet scientists really stop working on deadly pathogens, or did they simply learn to disguise them better? Officially, the Soviet Union fully supported the international ban on biological weapons. The USSR delegation actively participated in the negotiations on the convention. Soviet diplomats declared their commitment to the principles of humanity and the rejection of inhumane means of warfare. In practice, the signing of the convention did not stop the Soviet biological weapons program, but merely redirected it. Military research was disguised as medical and agricultural programs. A system of double standards was created, allowing international restrictions to be circumvented. The Interdepartmental Scientific and Technical Council on Molecular Biology and Genetics, established in 1974, played a key role in this process. Formally, the Council was responsible for coordinating fundamental research. In reality, it oversaw secret programs for the development of biological weapons. How was military research concealed under the guise of peaceful programs? Several standard cover stories were used. The development of combat strains of pathogens was carried out under the guise of vaccine creation. The study of means of delivering biological agents was disguised as work on aerosol therapy. A particularly effective cover was programs for protection against biological weapons. To create effective protection, it is necessary to know the characteristics of the enemy's potential weapons. This justified research into the most dangerous pathogens and methods of their combat use. The funding system was also modified to conceal the true purpose of the research. Military programs were funded through civilian ministries, health, agriculture, and higher education. Funds passed through several intermediary organizations, making it difficult to track their actual use. What new areas were developed as part of the covert program? The main focus was on genetic engineering methods for creating biological weapons. Recombinant DNA technology, which emerged in the 1970s, opened up fundamentally new possibilities for modifying pathogens. Soviet geneticists worked on creating chimeric viruses that combined the properties of different pathogens. In theory, such viruses could combine the high contagiousness of influenza with the lethality of Ebola, or resistance to treatment with rapid spread. A special area of focus was the development of binary biological weapons. Such systems consisted of two relatively safe components that only became lethal when combined in the victim's body. This made it possible to circumvent international agreements and control systems. At the same time, research was conducted on the creation of pathogens resistant to existing methods of treatment and prevention. Strains with multiple drug resistance were created, capable of overcoming the protection of antibiotics and antiviral drugs. How successful was the program to conceal military research? For a long time, Western intelligence agencies were unable to obtain reliable information about the continuation of biological programs in the USSR. Official statements by the Soviet leadership, backed up by show inspections of peaceful facilities, created the appearance of compliance with international agreements. The first serious suspicions arose in the early 1980s after an analysis of scientific publications by Soviet biologists. Some of the research was clearly military in nature, although it was disguised as fundamental science. The decisive factor was the defection in 1989 of GRU Colonel Vladimir Pasechnik, one of the leaders of the biological weapons program. His testimony revealed the true scale of the ongoing research and the system used to conceal it. It turned out that after 1972, the program had not only not been curtailed, but had expanded significantly. By the early 1990s, Dozens of scientific institutes, manufacturing enterprises, and military facilities were participating in it. Total funding reached billions of rubles annually. What biological agents were developed under the guise of peaceful programs? In addition to traditional bacterial and viral pathogens, Soviet scientists worked on fundamentally new types of biological weapons. The military applications of prions, toxins, and regulatory peptides were studied. Particular attention was paid to the creation of means of destroying crops and animals. Such weapons could cause famine and economic collapse in enemy countries without directly affecting humans. Formally, this did not fall under the prohibitions of the Biological Weapons Convention. Psychotropic biological agents were also developed, substances capable of altering human behavior and mental state. Such weapons could be used to disrupt enemy troops, suppress civil unrest, and carry out sabotage operations. How did the international community react to the revelations about the continuation of biological programs? The revelations of defectors caused a serious diplomatic scandal. The US and its allies demanded that the USSR completely cease military biological research and allow international inspectors access to classified facilities. The Soviet leadership was forced to admit to violations of the Biological Weapons Convention. In 1990, an agreement was signed on mutual inspections of biological facilities in the USSR and the US. A gradual winding down of the most odious military programs began. But was dual-use research completely stopped? Many programs continued in new forms. The names, sources of funding, and official research goals changed, but the essence of the work remained the same. The collapse of the USSR did not stop military biological research, but only decentralized it. Many programs came under the control of Republican governments. Some scientists emigrated, taking secret knowledge and technology with them. When the red flag with the hammer and sickle was lowered over the Kremlin in December 1991, it seemed that the Soviet Union's sinister military programs had disappeared into the past forever. But what happened to the gigantic arsenal of biological weapons and the thousands of scientists who had spent decades working on the creation of weapons of mass destruction? The collapse of the USSR presented the international community with unprecedented challenges in the field of biological security. Hundreds of laboratories, production facilities, and storage facilities containing dangerous pathogens were scattered across the territory of the former Soviet Empire. The system for controlling and protecting these facilities collapsed almost instantly. The first warning signs appeared as early as 1992. American intelligence services received information about attempts by criminal groups to sell samples of biological weapons. Offers to supply anthrax spores, plague cultures, and samples of particularly dangerous viruses appeared on the black market. How much biological weapons did the USSR accumulate over decades of secret programs? The exact data is still classified, but expert estimates suggest thousands of tons of ready-made biological agents. The arsenal included not only traditional pathogens, but also genetically modified strains unknown to Western science. Of particular concern was the fate of the core of the Soviet biological program, highly qualified specialists. By the early 1990s, tens of thousands of scientists, engineers, and technicians with unique knowledge in the field of biological weapons were working in the USSR. The economic collapse of the new independent states led to a catastrophic reduction in funding for science. Scientists' salaries were delayed for months, research programs were curtailed, and institutes were closed. Many specialists found themselves without work, and without a means of subsistence. Did these scientists become easy prey for terrorist organizations and hostile states? Western intelligence agencies received alarming reports about contacts between former Soviet biologists and representatives of Iraq, Iran, North Korea, and various terrorist groups. The most dangerous situation was with the senior staff of biological programs. These people knew not only the technology for creating biological weapons, but also the entire system for organizing such work, from scientific research to industrial production and combat use. To prevent a brain drain, the U.S. initiated the Nunn-Lugar program, aimed at employing former Soviet weapons scientists in peaceful projects. Russian, Ukrainian, and Kazakh scientists were offered contracts for research in the fields of medicine, agriculture, and ecology. How effective were these programs? Over the 20 years of the Nunn-Lugar program's existence, several thousand former developers of weapons of mass destruction were successfully employed. But this is only a fraction of the total number of specialists who possessed dangerous knowledge. Many scientists emigrated on their own, without waiting for international assistance. They found jobs at universities and pharmaceutical companies in the US, Western Europe, and Israel. Formally, they were engaged in peaceful research, but their knowledge could also be used for military purposes. Specialists who remained in Russia and other post-Soviet states posed a particular problem. Many of them continued to work in renamed institutes that had officially switched to civilian research. But were all military programs really shut down? An analysis of Russian scientific publications from the 1990s and 2000s shows that dual-use research continued. Work on the creation of new vaccines often included the study of the pathogenic properties of microorganisms. Research on biological defense required knowledge of the characteristics of potential biological weapons. The condition of biological material storage facilities was a cause for serious concern. Many of them remained without adequate security and control. In 1993, there was an attempted robbery of a warehouse in Sergiev Posad, near Moscow, where particularly dangerous viruses were stored. How much biological weaponry was destroyed after the collapse of the USSR? Official data indicates that most of the arsenal was eliminated under international control. However, experts doubt the completeness of the information provided. Some of the materials may have been hidden or taken away to an unknown location. Mobile biological laboratories, created during the Soviet era for sabotage operations, posed a particular threat. These compact installations could produce biological agents almost anywhere. After the collapse of the USSR, the fate of many such systems remained unknown. The problem was further complicated by the fact that a significant part of the Soviet biological arsenal was located not in Russia, but in other republics of the USSR. Ukraine, Kazakhstan, Uzbekistan, and Georgia inherited large biological facilities with unclear research status. Could terrorists have gained access to Soviet biological weapons? In the mid-1990s, the FBI investigated several cases of biological materials of unknown origin being offered for sale. Expert analysis showed that some samples could indeed have been produced in Soviet laboratories. The most high-profile case involved the Japanese cult, Aum Shinrikyo. The investigation established that representatives of the cult had repeatedly visited Russia and attempted to gain access to biological materials. Although no direct evidence of successful transactions was found, the very fact of such contacts caused serious concern. The current situation with the legacy of Soviet biological programs remains uncertain. Can we assume that the threat of biological weapons leaking from the former USSR has been completely eliminated? Many experts answer this question in the negative. The continuing secrecy of the Russian authorities, limited access for international inspectors, and the unclear fate of many former Soviet specialists all raise questions about the completeness of the elimination of the biological legacy of the Cold War. Today, 30 years after the collapse of the USSR, research centers that were once at the heart of the Soviet biological weapons program continue to operate in Siberian cities. Officially, they are engaged in peaceful research, the development of vaccines, diagnostic systems, and medicines. But can we be sure that new military developments are not hidden behind the facade of medical programs? The Vector State Research Center of Virology and Biotechnology in Koltsovo remains one of the largest virology centers in the world. Today, more than 2,000 employees work here, conducting research in several dozen areas. Officially, the center has been completely repurposed for peaceful purposes. What does the modern Vector do? Its main areas of work include the development of vaccines against particularly dangerous infections, the creation of diagnostic test systems, and fundamental research in the field of molecular virology. The center actively cooperates with international health organizations. During the COVID-19 pandemic, Vector became one of the key centers for studying the new coronavirus. It developed the Epivaccorona vaccine and created test systems for diagnosing the disease. The center's work has received widespread coverage in the Russian and international media. However, some aspects of Vector's activities raise questions among international experts. The center continues to work with collections of particularly dangerous viruses, including samples that may have dual-use potential. Many studies remain highly classified. What viruses are being studied at the modern Vector? Officially, the center works with pathogens that cause hemorrhagic fevers, smallpox, influenza, and coronavirus infections. New diagnostic systems are being created for the rapid detection of dangerous pathogens. Emergency prevention measures for biological threats are being developed. Particular attention is paid to research in the field of synthetic biology, a field that opens up fundamentally new possibilities for the creation of artificial biological systems. These technologies can be used for both medical and military applications. In recent years, Vector has been actively developing international cooperation. The center participates in World Health Organization programs and cooperates with research organizations in the US, Europe, and Asia. But isn't this cooperation a way to gain access to foreign biotechnologies? An analysis of scientific publications by Vector employees shows the continuation of research that may have military applications. The mechanisms by which viruses evade the immune response are being studied, strains with modified antigenic properties are being created, and delivery systems for biological agents are being developed. The Omsk Research Institute of Natural Focal Infections also continues to be active. Officially, the institute is engaged in epidemiological surveillance of particularly dangerous infections in the Siberian Federal District. Diagnostics and prevention methods for plague, tularemia, and anthrax are being developed. What new areas are being developed at the Omsk Institute? Current research includes studying the mechanisms of drug resistance in pathogenic bacteria. New antibacterial drugs are being created that are effective against resistant strains. Particular attention is being paid to the development of rapid diagnostic tools for biological threats. Portable systems are being created that can detect the presence of dangerous pathogens in various environmental objects in a matter of minutes. The Irkutsk Anti-Plague Institute is continuing its research into natural foci of plague in eastern Siberia and the Far East. The impact of climate change on the activity of natural foci of particularly dangerous infections is being studied. Predictive models of the epidemic situation are being developed. Do modern studies pose a threat to international security? Officially, all work is carried out within the framework of biological safety programs, but many areas of research have dual-use potential. The international community is particularly concerned about the secrecy that surrounds many Russian biological facilities. Access for foreign inspectors is limited, and information about the nature of the research is provided selectively. In recent years, the Russian authorities have been actively developing programs in the field of biological protection. New radiation, chemical, and biological protection troops are being created. Means of detecting and neutralizing biological threats are being developed. Is the biological defense program a cover for the resumption of offensive research? The line between defensive and offensive programs in the field of biological weapons is extremely blurred. To create effective defenses, it is necessary to know the characteristics of the enemy's potential weapons. Modern Russian military doctrines include the concept of new generation biological warfare. It is assumed that biological agents will be used not only against people, but also against crops, domestic animals, and the enemy's ecosystems. Research is also being conducted in the field of psychotropic biological agents, substances capable of affecting the psyche and behavior of people. Such agents can be used to disrupt enemy troops, suppress civil unrest, and conduct special operations. What role do private biotechnology companies play? The private biotechnology sector is actively developing in Russia. Many companies have close ties with state research centers and the military. Some private firms receive government contracts to develop biological defense agents. The nature of this work is often classified, which makes it difficult to monitor compliance with the Biological Weapons Convention internationally. Of particular concern is the development of Russian programs in the field of artificial intelligence as it applies to biological research. AI can significantly accelerate the process of creating new pathogens or means of delivering them. Does Russia cooperate with other countries in the field of dual-use biological research? There are known contacts between Russian scientists and their colleagues from China, Iran, and North Korea, countries suspected of developing biological weapons. The current situation in the field of biological security remains extremely uncertain. The lack of transparency in Russian programs, the continuing secrecy of many studies, and the development of new dual-use biotechnologies pose serious risks to international security. In the era of globalization, secrets know no borders. The same applies to the technologies for creating biological weapons developed in Siberian laboratories. After the collapse of the USSR, this knowledge began to spread around the world, finding new applications in the most unexpected places. Have Russian biotechnologies become a commodity on the international market of death? The first signs of the export of Soviet biological technologies appeared as early as the beginning of the 1990s. The economic crisis forced Russian research centers to look for new sources of funding. International contracts became the only way for many research programs to survive. Formally, this was peaceful cooperation in the field of medicine and biotechnology. Russian institutes offered their foreign partners their developments in the field of vaccine creation, diagnostic systems, and biopharmaceuticals. But were these technologies always used exclusively for peaceful purposes? The countries of the so-called Axis of Evil, Iran, Iraq, and North Korea showed particular interest in Russian developments. These states were actively developing programs to create weapons of mass destruction and needed advanced biotechnologies. Iran became one of the largest buyers of Russian biological technologies. Officially, cooperation was carried out within the framework of programs to develop the pharmaceutical industry and healthcare system. In practice, Iranian specialists gained access to technologies for working with particularly dangerous pathogens. What specific technologies were transferred to the Iranian side? It is known that equipment for working with viruses under conditions of maximum biological protection was supplied. Russian specialists trained their Iranian colleagues in methods of cultivating and concentrating pathogenic microorganisms. Particular attention was paid to the transfer of technologies for creating aerosol forms of biological agents. This knowledge was formerly intended for the development of inhaled drugs. But the same technologies could be used to create biological weapons. North Korea also actively cooperated with Russian biologists. The Democratic People's Republic of Korea was primarily interested in technologies for the mass production of biological agents and methods for their preservation for long-term storage. Such knowledge is critical for the creation of strategic reserves of biological weapons. Were former developers of Soviet biological weapons involved in these deals? Many Russian scientists who had worked in classified programs found themselves without work and means of subsistence after the collapse of the USSR. Offers from foreign customers became the only way for them to apply their knowledge and skills. There are known cases of former Soviet biological weapons developers being directly involved in foreign programs. They acted as consultants, teachers, and research project managers. Their unique experience was highly valued by customers. China chose a different strategy for obtaining Russian biotechnology. Instead of direct purchases, Chinese companies actively invested in Russian biotechnology firms, thus gaining access to advanced developments. What Russian biotechnologies were of particular interest to the Chinese side? The priorities were genetic engineering technologies, methods for creating recombinant strains of microorganisms, and biological defense systems. China sought to eliminate its lag in the field of military biotechnology. Scientific cooperation also developed through a system of joint research programs. Chinese graduate students and interns were sent en masse to Russian biological institutes. Formally, they studied the fundamentals of biology. In practice, they gained access to classified technologies. Private intermediary firms played a special role in the export of biological technologies. These companies served as an intermediary between Russian developers and foreign customers, making it difficult to track the actual destinations of shipments. How did Western countries react to the spread of Russian biotechnology? The U.S. and its allies attempted to control the export of dual-use technologies through a system of international sanctions and export restrictions. However, the effectiveness of these measures remained limited. The problem was complicated by the fact that the line between peaceful and military biotechnologies is extremely blurred. Equipment for the production of vaccines can be used to grow combat strains of pathogens. Diagnostic systems can be easily modified to detect biological agents. In the 2000s, Russian biotechnology exports took on a more civilized form. Joint ventures, technology parks, and research centers were created, but the essence of the process remained unchanged. Russian knowledge in the field of biological weapons continued to spread around the world. What role did international terrorist organizations play in this process? No direct evidence of cooperation between Russian scientists and terrorists has been found. However, there are known cases of attempts by representatives of extremist groups to obtain biological materials. The possibility of creating a decentralized network of biological terrorism is of particular concern. The spread of Russian technologies could lead to the emergence of numerous independent centers for the production of biological weapons around the world. The current situation with the export of Russian biotechnologies remains extremely uncertain. Is the transfer of dangerous knowledge and technologies to foreign partners continuing? The answer to this question is complicated by the secrecy of many international contracts. The development of digital technologies is creating new channels for the dissemination of biological knowledge. Secret developments can be transferred via the Internet in the form of computer programs, databases, and virtual laboratories. It is virtually impossible to control such information flows. What threat to international security does the continued export of Russian biotechnology pose? The spread of biological weapons technology could lead to global destabilization of the international security system. The emergence of new biological powers will change the balance of power in the world. Today, in the 25th year of the 21st century, humanity stands on the threshold of a biological revolution. Advances in genetic engineering, synthetic biology, and artificial intelligence open up limitless possibilities for manipulating living matter. But won't these same technologies become the basis for creating a new generation of biological weapons? Modern biotechnology has radically changed the possibilities for creating pathogenic organisms. What used to require years of research and millions in funding can now be done in a small laboratory in a matter of weeks. CRISPR technology allows genes to be edited with scalpel-like precision. What new threats do modern biotechnologies pose? First of all, it is the possibility of creating artificial pathogens that do not exist in nature. Such organisms can have predetermined properties: high contagiousness, resistance to treatment, the ability to affect certain organs or systems. The development of technologies for resurrecting extinct pathogens poses a particular danger. Scientists have already reconstructed the genome of the 1918 Spanish flu virus, which killed tens of millions of people. What will prevent terrorists from using the same methods to recreate other historical pandemics? Artificial intelligence is becoming a powerful tool for biological design. Neural networks can analyze millions of genomic sequences, identifying the optimal combinations of genes to create pathogens with specific properties. The process of developing biological weapons is accelerating exponentially. The legacy of Siberian biological laboratories is becoming particularly dangerous in the context of modern technology. Decades of accumulated knowledge about pathogenic microorganisms can be used as a basis for creating a new generation of biological weapons. Are biological weapons development programs still ongoing in Russia? Officially, all such programs have been terminated. However, analysis of open sources shows that dual-use research is continuing. Technologies are being developed that can have both peaceful and military applications. Of particular concern is the Russian program to create a genetic passport for the nation. Officially, it is aimed at studying the genetic characteristics of the Russian population for medical purposes. But could this program be used to create ethnically specific biological weapons? Modern Russian military doctrines include the concept of new generation biological threats. Scenarios are being considered for the use of biological agents against the enemy's infrastructure, agriculture, and ecosystems. Biological warfare is becoming multi-level and multi-vector. Technologies for controlling behavior through biological agents are also being developed. Substances are being created that can affect a person's mental state, cognitive functions, and emotional sphere. Such weapons can be used to disrupt society without physically destroying people. What role do private biotechnology companies play in creating future threats? The commercial biotechnology sector is developing faster than government programs. Many private firms possess technologies that can be used for military purposes. The phenomenon of garage biology poses a particular danger: the emergence of affordable equipment that allows genetic experiments to be conducted at home. Biohackers are gaining capabilities that were previously only available to large scientific centers. The globalization of biotechnology complicates the control of the spread of dangerous technologies. Knowledge and materials circulate freely between countries through scientific publications, international conferences, and educational programs. Is the international community ready for new biological threats? Existing international agreements were created in the era of traditional biotechnology. They do not take into account the possibilities of modern methods of creating artificial pathogens. The international system for monitoring biological threats remains fragmented and ineffective. Many countries do not provide complete information about their research in the field of biosafety. There is no unified early warning system. The COVID-19 pandemic has exposed the vulnerability of the modern world to biological threats. In 2 years, a single pathogen has radically changed life across the entire planet. What will happen if the next pandemic is the result of the deliberate creation of biological weapons? Of particular concern is the possibility of creating a doomsday pathogen, an artificial organism capable of destroying all life on Earth. Modern technologies make such scenarios theoretically possible. What measures are needed to prevent a biological catastrophe? A radical modernization of the international system for controlling biological research is required. New agreements are needed that take into account the capabilities of modern biotechnology. The development of biological defense systems plays a crucial role. Technologies for the rapid detection of new pathogens, methods for the emergency creation of vaccines and medicines, and systems for mass prevention among the population are needed. International cooperation in the field of biosafety is also required. The threats are global in nature and can only be effectively prevented through the joint efforts of all countries of the world. The legacy of Siberian biological laboratories reminds us of the consequences that the militarization of biology can lead to. Will humanity be able to avoid repeating the mistakes of the past in the era of the biotechnological revolution? The answer to this question will determine the future of our civilization.